All right, so switching gears a little bit now, we're going to talk about cell signaling. So a couple key learning objectives for today. We're going to discuss different types of secondary messengers. We're going to go back over GTPases and the G protein coupled receptors, and then talk about a couple of signaling pathways that involve G protein coupled receptors and other secondary messengers. And then our disease topic today is cholera. So cholera, as you might know already, is an infectious disease. So this is like a posting from 1850s, around 1854, when there was this very notable cholera outbreak in the London, England area. So this is known as the Broad Street outbreak, and this was actually before germ theory. So the prevailing theory at the time was one of miasma, where there's decaying things that can transmit disease through the air. So that's what a lot of people thought was happening. It was a big mystery, and there was a physician named John Snow that actually tracked down epidemiologically where everyone who was being infected and dying from cholera lived and what similarities were between them. And they were able to trace it to this Broad Street pump, their water pump that they all got their water from. And so this helped solve the theory of what is causing cholera in this city. And there's actually a really great book about it called The Ghost Map, if anyone's interested. But there's been many other cholera outbreaks since then. So this is not just an ancient disease. This is a disease that still has different outbreaks occurring now. So a lot of times this is coming at times of instability in a country. So in Haiti, there's a really famous cholera outbreak because of its massive devastation in 2010 that came right after a really terrible earthquake with hurricanes following that summer. So the earthquake was in January and displaced millions of people. The infrastructure was destroyed. And then the subsequent hurricanes really put the country in a place of devastation that made it vulnerable to this cholera outbreak. So there was about 10,000 deaths that have accumulated from this, and there was a lot of challenge in eradicating cholera. Eradicating cholera following this. So they, in 2018, had declared that Haiti was cholera-free, but actually just last year there was a few cases. So this is still an ongoing battle in a lot of places. And similarly in Yemen, during this extensive period of civil war, there was the then greatest cholera outbreak in history. So at the time, Haiti had the greatest cholera outbreak, and then Yemen in around 2018 had a really devastating cholera outbreak that came at the time of civil war, again, because the infrastructure of the country was down and it was vulnerable. So cholera is a disease that is caused by this bacterium, Vibrio cholerae. So it's a waterborne disease, and it causes really, really severe and dangerous diarrhea. So the amount of devastation that this causes maybe can be clear from the extensive deaths that are caused in these outbreaks, but it is generally transmitted through contaminated water sources. So again, this isn't necessarily the case in a lot of places where there's there's sufficient sanitation in place, but in times when that sanitation might be at risk. So there's also various countries that have tried to weaponize cholera as a biological toxin, specifically by contaminating these water sources. So how this progresses, part of the reason why it's so dangerous is because this massive onset of diarrhea occurs very quickly. So in the most severe cases, this can lead to death on the order of hours. And this is often due to a person becoming very hypotensive. So does anyone want to volunteer what hypotensive is? Yeah. Low blood pressure, exactly, which makes sense from a lack of fluid. But more commonly, this disease progresses over the course of hours to days. and leads to just a severe volume loss in terms of the amount of water that is excreted from the volume. So this is known as hypovolemic shock. So ultimately, the cause of death is dehydration, which is, again, this lack of water. But if the main treatment then is replacing these fluids, so there can be a period of recovery, unlike some of the other diseases we talked about, where if you get infected with cholera, then adequately replacing the fluids that are lost can lead to recovery. So part of the reason why it's important to talk about today and where it ties in is because this toxin that the cholera secretes is a G protein modifier. So we'll come back to exactly how this fluid loss occurs at the end of the lecture. Okay, before we get started, I know there's a lot of pre-lecture material. Is there any outstanding questions about that? We'll kind of circle back on some of those ideas in the lecture, but just wanted to see if there was any questions. Okay, hopefully it was helpful, and feel free if any questions about the pre-lecture come up throughout the course of this, to pop in at any point. So G proteins are this heterotrimeric protein complex that are associated with a receptor. So the receptor is GPCR, which stands for G protein coupled receptor. So these are receptors that are specifically causing a change in the G protein. And this is a really important class of receptors and proteins to study, because there's over 900 of them in your body. This is also a really important signaling pathway that is targeted pharmacologically. So there's a lot of companies that are investigating ways to target these G protein coupled receptors in drugs. But a lot of these are actually involved in smell and taste receptors. So they're littered throughout your body. And how the signaling pathway works is you have this 7 helical protein motif that passes through the membrane. So it's a transmembrane protein. It has an affinity for a specific ligand. So that's this X in this case. And when the ligand binds, it's going to change its conformation. And this conformational change of the receptor is going to then be transmitted to this G protein complex. So we have this alpha, beta, and gamma subunit here. The gamma is actually also bound to the membrane, as the alpha is here. But this beta gamma complex is transferring the conformational change to the alpha subunit. So the alpha subunit is bound to GDP in its inactive state. So the alpha subunit of this protein is a GTPase. So it's an enzyme, and it binds GTP and hydrolyzes it. So this is something we've heard before in terms of these proteins and enzyme activity. And so when the beta gamma complex of the G protein changes its conformation, it also changes the conformation of the alpha subunit and encourages it to release the GDP. So once it releases GDP, then it's free to bind GTP, and that activates it and changes it conformationally so that it can dissociate from the other portion of the beta gamma G protein. So once it's bound to GTP and it's free from the rest of the G protein complex, then it can diffuse laterally across the cell membrane. And it'll do this until it finds its target. So in this scenario, this alpha subunit is activating this enzyme adenylyl cyclase. So that's what the AC stands for here. And this is another enzyme that is also bound to the cell membrane. So this activation of adenylyl cyclase will turn on this enzyme activity, and the substrate for adenylyl cyclase is ATP. And so what adenylyl cyclase does is take ATP, remove 2 of the phosphate groups, and turn it into this cyclic molecule. So that's kind of where the cyclase comes from, and that cyclic molecule is referred to as cyclic AMP. So there's now a monophosphate. Oops. And it is a cyclic molecule. So the structure of cyclic AMP, I believe, is in the pre-lecture, and this is one of those secondary messengers that's going to be important in G protein coupled signaling as well as in other pathways. So because adenylyl cyclase is an enzyme, as long as it's active, it can continue to take in its substrate, which is ATP, and continue to produce more cyclic AMP. So this part of the signaling cascade is going to be amplifying the signal. So this is another aspect of the secondary messengers, is that they're often amplifying signal. And this is all happening in the cytoplasm. So the production of cyclic AMP will be the product of cyclic AMP will be free to diffuse all around the cell, and then it can hit any of its target proteins that it will have some activity in. So this is occurring as long as adenylyl cyclase is active. And so because this is such a potent signaling pathway, there has to be some negative regulation on it. And so there's a couple layers of this. One of them is that this alpha subunit, since it is itself an enzyme, after some amount of time, it will hydrolyze the GTP that's bound to it. So it'll undergo its enzymatic activity and hydrolyze GTP to GDP, and that will return it to its inactive form. So what that does is it kind of uses this alpha subunit as like a molecular timer. So the activity of this alpha subunit will determine how long it'll take for that GTP to be hydrolyzed and the signal to be continuing to stay on. So once the alpha subunit hydrolyzes GTP, then it will no longer be activating adenylyl cyclase. So that means that adenylyl cyclase will no longer be catalyzing this transition of ATP to cyclic AMP, and it'll stop producing the secondary messengers. But this other layer of regulation comes with the fact that there's already a bunch of cyclic AMP now present in the cytoplasm. So as long as the cyclic AMP is around, then it'll keep progressing that signal. And so the second layer of regulation is around degradation of the cyclic AMP. So that is through this phosphodiesterase. And so this uses cyclic AMP as its substrate, and it deactivates it from its secondary messenger. All right, any questions about this? Yeah. Yeah, it is regular AMP, right?
Yeah.
Yeah.
There's a ester bond, there's a phosphoester bond that kind of promotes like the stabilization of cyclic AMP. And so it clips that and this becomes regular AMP. Then it's one phosphoester bond.
Yeah, good question. All right. So what does this, that was a bunch of cartoons, schematics of how this all works. But in terms of what this protein actually looks like, you can see visualized here. I think this is a video. So this is the 3 subunits of the G protein. So this will be associated with the membrane, specifically at the alpha and gamma subunits. And you can kind of see here on the inside of this alpha is that GTP binding pocket. So this is the unit that's specifically binding GTP. And this alpha subunit can come in a variety of flavors. So there's a big family of G proteins. They're all acting as GTPases, but the activity of the alpha subunit is going to be different in different versions. So it can act on different enzymes other than adenylyl cyclase. It can be inhibitory rather than activating. And we'll give a couple of examples of that in the lecture. All right, so that structure was experimentally determined in 1996 and was a big progression for the field. but it's significantly harder to get the structure with an associated receptor. So this paper in 2011 was able to determine that experimentally. So this is an example of a G protein coupled receptor and how it's bound to the G protein. So in this case, it's the beta adrenergic receptor. And so this is going to be transmembrane, like we said, with 7 alpha helices that pass through the lipid bilayer, and then it'll be associated with the G protein on the other side. All right, so looking downstream from where we just talked about, we got all the way to cyclic AMP. And so what happens after cyclic AMP? So one of the main targets of what cyclic AMP does in the cell is turn on protein kinase A. So PKA, or protein kinase A, stands for CMP activated protein kinase. So the activity is in the name, is when there's an abundance of cyclic AMP in the cytoplasm, and that'll activate this PKA. And this is a kinase, so it's going to be adding phosphate groups to different types of proteins. And what this looks like is shown in this schematic here. So we both have a cartoon diagram as well as the actual molecular structure of this. And it's comprised of 2 catalytic subunits and 2 regulatory subunits. So the catalytic subunits are shown here in white, and the regulatory subunits are composed of these 2 different domains. So this green is one domain and the blue is another domain. These are cyclic nucleotide binding domains is what this stands for. And this is because they're binding directly to cyclic AMP. So in order to activate protein kinase A, you have to have cyclic AMP come in and bind each of these domains of the regulatory subunits, and then it'll disassociate from the rest of this protein complex and leave this catalytic domain active and available to bind ATP and phosphorylate other proteins. So this, in between this N lobe and C lobe, is specifically where this catalytic domain is. So this schematic just shows, this is kind of like a side view and a top view, and the same in the protein structure. All right, any questions about this so far? Yeah. I would guess, but I'm not exactly sure. I'm not sure if it's on here exactly. Yeah, I would suspect that that's probably where the N terminus and the C terminus lies, but good question. All right, we'll keep chugging along. So we talked a lot about cyclic AMP, but there is a lot of other types of secondary messengers. So we're going to walk through a couple of signaling pathways that demonstrate how these secondary messengers come into play. And so specifically, we're going to talk about 2 that are derived from lipids. So in this case, we have this enzyme phospholipase C that is catalyzing the emergence of these 2 secondary messengers. So can anyone guess what a phospholipase does? Yeah. Exactly. Yeah. So it catalyzes the breakdown of phospholipids. So it cleaves a phospholipid specifically. And the one that it cleaves is this 4,5 biphosphate, this PIP2. So this is the acyl chains embedded in the lipid bilayer. And then we have the glycerol, the phosphate group connected to the inositol ring. And there's 2 phosphate groups here. And so what this PLC enzyme does is it comes and cleaves right here at the end of the glycerol and leaves this inositol group with 3 phosphates. So this is IP3, or inositol triphosphate. And then the glycerol connected to the 2 acyl chains. So diacylglycerol. This might be familiar when we talked a couple lectures ago about this. So in this case, both the diacylglycerol and IP3 are serving as second messengers, and we'll show exactly how that works in the next slide. Okay, so like I said, these pathways that we're going to walk through are actually going to be specifically relevant to smooth muscle. So these are the muscles that are lining different organs. So you can think like your blood vessels or your esophagus or other things. So in this pathway, we're going to start with angiotensin 1. And so this is a vasoconstrictor. So this is going to cause the activity of these smooth muscles. And its receptor is a G protein coupled receptor. So when you have angiotensin in the bloodstream and it's able to bind its G protein coupled receptor in smooth muscles, then that's going to cause this conformational change in the G protein coupled receptor. And it's then transmitted to this G protein complex. So similar to before, it's going to transmit this conformational change to the beta gamma unit, which is going to allow the dissociation of GDP from the alpha subunit, activating it, allowing it to bind GTP, and then diffuse along the lipid bilayer. But in this case, the target of the alpha subunit is this PLC enzyme. Oh man, it's really turning off a lot today. Let's see. All right, there we go. So this is that phospholipase C, which is going to cleave this PIP2 phospholipid in the cell membrane. And again, this is going to expose these two secondary messengers. So this is the IP3 and the diacylglycerol. So the diacylglycerol will continue to be bound in the lipid bilayer because these acyl groups are hydrophobic. They're wedged in this hydrophobic region of the cell membrane, but the IP3 is going to be free to diffuse. So it's no longer bound to the phospholipid, and it'll diffuse freely all throughout the cell. And so a target for this IP3 is the IP3 receptor, and this is expressed on the smooth ER of those cells. So the smooth ER can serve as a major storage for calcium ions in the cell. So the concentration of calcium is much higher in the smooth ER, and this receptor, when IP3 binds to it, opens a channel that allows the calcium to diffuse into the cytoplasm. So we talked a little bit before about in smooth muscle, and in both smooth and striated muscle, the abundance of calcium activates contraction. So in this case, you have the activation of the myosin light chain kinase that's going to allow the contraction through activation of this enzyme and the subsequent contraction of the muscle. All right, so this is turning this signal on, and so we don't want these muscles to be active all the time or constitutively active. So there's going to be some negative regulation here, and so we talked about with the phosphodiesterase breaking down cyclic AMP. There's also a negative regulation in this pathway, so that's going to start with this enzyme protein kinase C. So we talked about protein kinase A before. And so this is going to be already present in the cell cytoplasm. And in the presence of calcium, it will bind calcium, and then it'll also bind the diacylglycerol, and this will activate its activity. So it needs both of these secondary messengers binding in order to activate this, PKC enzyme. And this will then phosphorylate a bunch of different types of enzymes in a complex system that eventually leads to the inhibiting of contractility or stopping the contraction of the muscles. Yeah. That's a good question. I think that some of the downstream targets for PKC eventually close that channel, but I'm not sure what they are off the top of my head.
Makes the PKC more empathic, but help, there's all these channels have different there's always a regulatory feedback mechanism that makes sure that we shut off these channels, that we're not always either dumping calcium into the cytoplasm, or there's another slide we'll talk about calcium being shuttled into the cell from the competitor parts. And so PKC, active PKC helps to shut down that channel so that we don't constantly have calcium being shuttled from the smooth ER into the cytoplasm.
Yeah, good question. Okay, so this slide is just bullet points that kind of summarize what we just went through. So we'll walk through it one more time. Hopefully this is helpful in studying. But this G protein coupled receptor activates this heterotrimeric G protein. So the alpha subunit of it is going to activate phospholipase C. And so this phospholipase is going to cleave this phospholipid that's embedded in the membrane, PIP2, and it's going to yield these two secondary messengers, so diacylglycerol and IP3. And so the IP3 is going to diffuse, bind to the IP3 receptor on the smooth ER, and release calcium. Then on the negative side, the calcium and the diacylglycerol together are going to activate this PKC enzyme, and this is occurring at the cell membrane. And this leads to the eventual pumping of calcium back into the ER to restore this stock. And the diacylglycerol will be phosphorylated and remetabolized into the bilayer. Any questions about this? Yeah. The diacylglycerol is necessary to activate PKC. So even though it's staying at the membrane, it's still interacting with that protein to cause the activation of it. So that's like the whole concept behind these secondary messengers, is that they're required for the action of the next step in the signaling cascade. So that's why diacylglycerol would be considered a secondary messenger, even though it's not diffusing freely like a lot of secondary messengers do. It's required in this case to activate this other enzyme.
For PKC, one analogy that I'll kind of helpful is it's kind of like two-factor authorization when you log into your emails or anything that you get. PKC requires two-factor authorization. It requires calcium and it requires diacylglycerol. And so like that's like in this system, like that's the main function of diacylglycerol, to serve as one of the activating factors for PKC.
Thanks. It's a good analogy. All right, moving on to another signaling pathway. So this is also involved in smooth muscle, but this is more leading to relaxation rather than contraction. So this is a guanylyl cyclase signaling pathway. And this is different in the sense that this technically isn't a G protein coupled receptor. So in the case of a lot of these cells, like we were talking about, there's different transporters, both on the smooth ER, like we mentioned, but also on the cytoplasm that will help restore some of these stocks of calcium that are being depleted through the binding of different kinases and regulatory molecules. So in contraction, you have the production of nitrous oxide. So this is another one of these secondary messengers. This is a gaseous molecule, and so it's free to diffuse. It can be produced in cells and responded to in those cells, but oftentimes it's produced in a cell and then diffuses to neighboring cells and creates some activity in those neighboring cells. So in this scenario, you can imagine there's a neighboring cell, it's producing a lot of nitrous oxide, and that's going to diffuse to another cell that can freely pass the lipid bilayer and activate this enzyme guanylyl cyclase. So this is similar to adenylyl cyclase, like we mentioned, except the substrate now is GTP instead of ATP. So the associated nucleotide there. So when guanylyl cyclase is activated, it'll take GTP and it'll transfer it to cyclic GMP, similar to cyclic AMP. And so the cyclic GMP can also act as a secondary messenger, activate another enzyme known as PKG, similar to how cyclic AMP can activate PKA. So a lot of the terminology here kind of explains itself as long as you know what it stands for. And this cyclic GMP bound PKG is going to activate this enzyme to, again, utilize its kinase activity and phosphorylate a lot of different proteins in the cell. So some of the downstream activity of PKG that leads to muscle contraction is the phosphorylation of these lipid bilayer bound calcium channels. So the phosphorylation of this channel will close it and stop the influx of calcium ions into the cell. And it'll also phosphorylate a pump that is also on the smoothie R that's shuttling calcium back in. So this is depleting the calcium ion concentration in the cytoplasm and restoring the calcium ion concentration in the smoothie R to effectively contract muscle or to relax muscles, to stop the contraction. All right, so again, some bullet points just walking through this process that's hopefully helpful to come back to later. So this nitrous oxide diffuses to its target. So this is technically a reactive oxygen species. It's gaseous. It doesn't need any transporters or receptors. It can just bind or diffuse right through the lipid bilayer. And its target here is guanylyl cyclase. So it activates guanylyl cyclase. It's an agonist. If it was inhibiting, it would be an antagonist. And this guanylyl cyclase produces cyclic guanosine monophosphate, or cyclic GMP. This cyclic GMP then acts on protein kinase G, which is again GMP activated protein kinase. And then this is a kinase that's going to phosphorylate a bunch of substrates, including these two different channels that will effectively lower the calcium ion concentration in the cell. So similar to the adenylyl cyclase pathway that we talked about in the beginning, cyclic GMP has a negative regulator of phosphodiesterase 5. So again, a phosphodiesterase that'll cleave the ester bond and produce GMP. Any questions about this? I know it's a lot of acronyms. I think that's one of the tricky parts about this part of the class is a lot of these signaling cascades are just a series of acronyms. But thankfully, in this case, a lot of them are these enzymes where if you know what the name of the enzyme is, you can figure out what the enzyme is doing. So hopefully with that knowledge, you can just seek to memorize not just what the acronym and activity is, but what the name is that the acronym stands for, and that can help you remember its activity. So that would be my advice for studying these pathways, is remembering what the acronym stands for so that you can then remember what the activity of the protein is.
One other thing that's also helpful is that, we're reaching a point where we're kind of building, this is always the case, but we're building upon what we've learned in previous lectures. So if you think back to what might feel eons ago, when we talked about molecular motors and we talked about how on the regulatory light chain of myosin, there's a non-striated muscle, that there's a phosphorylation event that happens that's regulated by calcium. That calcium that we talked about in that lecture, and if you're not remembering it, you can go back and listen to it, because it was a great lecture. But if you go back and listen to it, we talked about this intracellular calcium concentration and the role it plays in non-striated muscle and smooth muscle. That's what we're talking about here. And the first pathway was all about smooth muscle cell contraction. And so the shuttling in of calcium into the cytoplasm it's all tied together. What we talked about back then with myosin, as well as what we're talking about here, and the secondary messengers that are all part of the same process. And this nitric oxide guanylyl cyclase pathway is all about smooth muscle cell relaxation. You can't always have your smooth muscle cells, particularly around your large arteries, always contracting. You need to also enable their relaxation. So that's kind of like the big picture as it relates to previous content, but also the big picture of these two pathways that we've been looking at.
Thanks. Yeah, it's good in terms of big picture, understanding what the role of these secondary messengers are doing, how they're transmitting signals from one protein to another, and then what the ultimate end goal of these pathways is. Okay, so then what does this have to do with cholera? Getting back to our disease topic, we mentioned before that cholera targets the alpha subunit. So in particular, the G protein coupled receptor that it's acting on is in the intestinal lumen, so in your gut. And this G protein coupled receptor has a ligand of vasoactive intestinal peptide, so or VIP. And so when this binds its receptor, the ultimate goal is to act on the vasodilation. So similar to before, the binding of the ligand is going to cause this conformational change. It's going to be eventually transmitted to the alpha subunit, and this is going to activate it, transfer it to adenylyl cyclase to activate adenylyl cyclase, creating cyclic AMP, activates PKA, and PKA in this case, one of the targets is this channel, this ion channel that leads to the intestinal lumen, and the phosphorylation of this ion channel opens it up conformationally, allowing the passage of chlorine. So this CFTR stands for cystic fibrosis transmembrane conductance regulator, which is a long title, but it has been implicated in cystic fibrosis because this transmission of ions into the lumen or in other kind of linings raises the osmotic pressure, and eventually, and the increase in osmotic pressure or number of solutes is going to drive the transfer of water. So this is important physiology, normal and healthy, and in cystic fibrosis, when this transporter is non-functional or partly non-functional, then the lack of transmission of these ions leads to a lack of hydration of these mucosal membranes, eventually leading to the number of symptoms that are associated with cystic fibrosis. So in this case, the end goal of this pathway is raising the solute concentration in this intestinal lumen space, which drives the migration of water also into this space. Okay, so the cholera toxin is acting specifically on the alpha subunit of this G protein, and it does so through adding this ADP ribosylate group. So the binding site is here, the active site for the alpha subunit is here. So it's not acting exactly at the active site, but what this change does is that it takes away the enzymatic activity of this alpha subunit. So it can no longer hydrolyze GTP. Yes. So this entire structure here is the alpha subunit. This little red piece down here is where it binds GTP. And then this place in yellow is where the ADP ribosylation occurs. Yeah. Yeah, so this is a structure, so it's ADP, so you have the adenylyl, the phosphate, or two phosphates, you have the ribose that's in ADP, but then at the end of the phosphates is another ribose. I actually put the structure in here because I figured somebody might ask. So this is the structure of ADP ribose. This isn't something you have to know, but it's basically adding this to a portion of the protein. Does that help? Okay. Okay, so again, this is inhibiting the enzymatic activity of the alpha subunit so that it can no longer hydrolyze GTP. So does anyone want to guess at what that's going to do for the signaling pathway? Yeah. Exactly. Yeah, it stops that negative regulation, in particular, what we were talking about with this molecular timer. So because of the eventual enzymatic activity of the alpha subunit, it will disassociate with the adenylyl cyclase and stop this cascade. But with this modification, if it is always bound to GTP, then it will never deactivate. So downstream, what happens? You have an always active alpha subunit, and so this will be continually acting adenylyl cyclase, producing a lot of cyclic AMP that's going to act on pKa and this downstream target of this chloride channel. And so the shuttling of chloride out into this intestinal lumen will again raise this solute pressure, and it will drive up the amount of water. And so this is what's happening in cholera, is you just have massive amounts of water leaving your tissue, going into your intestinal lumen, and then eventually being excreted. Any questions about this? Yeah. If all of the chloride ions are in the intestinal lumen? That's a good question. I think so. There's chloride that's existing in a lot of parts of your body. So this is specifically referring to the shuttling of chloride from the cell. So I imagine there's other channels that are restoring it so that membrane potential can stay around the same. So I think that that wouldn't be the limiting factor in this case. The byproducts of losing so much water would be devastating before chloride ions would run out, I believe.
One of those that's helpful when thinking about how ions move across the cell membrane or how water follows. This also helps with like when we talk about hydration later. When chlorine keeps being shuttled out, and this is true for cystic fibrosis, that's like the same channel that prepares the snake. It's a chlorine ion transporter. When chlorine is shuttled out, sodium will typically follow chlorine. There's other transports that help mediate that, but typically on average, sodium will on average follow where chlorine goes. And as that starts to happen, you're creating this osmotic pressure where water, there's not like a transport for water. And so while ions have these transporters, water is not going through some sort of transporter. There's a osmotic pressure that drives water through these tight junctions between cells. That water is then going to the intestinal lumen. And as this is happening, this is happening so much, like this ADP orb oscillation is constantly creating activity here. This is how, for example, in cholera, people can lose about, in the form of, it's a little disturbing, but it's the reality. In the form of diarrhea, these patients are losing about a liter of water per hour. Like that's how quickly this is happening and how severe all this is happening.
Yeah, no, it's good. I appreciate you adding that. Yeah, so because there's so much water loss, like we were talking about before, the main treatment approach is restoring that water through hydration, which makes sense, right? But a lot of times in the scenarios where cholera outbreaks are happening, this is often due to contaminated water sources. So then this becomes a little bit more tricky than you might expect. So because this has been a global problem in a number of countries, the World Health Organization has developed this quick fix for dehydration. So the purpose of this is to just maximize the amount of water uptake. And so this makes sense if you think about electrolytes or Gatorade. The purpose is to increase the amount of water uptake in your body. And so this is just a half teaspoon of salt, eight teaspoons of sugar in 1 quart of water. And this is because the sodium as well as the chloride will be transported together, like we were talking about. And the sodium transport is actually a sodium glucose symporter, which means that they're transported in the same direction at the same time. So having both of these together will increase the amount of overall transport. It'll also increase the amount of solute concentration in the gut. And so this will again drive the osmotic pressure, hopefully in the other direction, so that your tissues have the water that they need. All right. So with that, we'll just finish up with a couple people that are working on this. So at UVA, we have a lot of great infectious disease researchers. So Richard Grant, or Dick Grant, is internationally recognized in enteric infections. He actually helped found the Global Center for Global Health here at UVA. I think they changed names recently, but they do a lot of great work. So he's in the Division of Infectious Diseases. But then there's also a lot of people working on different types of second messenger signaling. So Jeff Sosserman is one who works specifically with cyclic AMP signaling in the heart, and Craig Newmaker also in pharmacology. So with that, I hope you guys have a great spring break. Happy Leap Day, and I'll see you next week.